Right, so we're beginning today. Well, this is biotechnology in medicine, but we will be beginning today with regenerative medicine, and then we'll also be speaking about some genome editing and human genome editing. Let's have a discussion on that. But in case you do not know, regenerative medicine seeks to replace or regenerate damaged or diseased tissues and organs. So that would include stem cell therapy, tissue engineering, and 3D bioprinting. And that offers promising approaches to repair and restore damaged body parts, and also addressing the shortage of um, donor organs. So I'm not sure if you're aware, but especially in the US, there is a long list of people waiting to get organs. So with this technology, we may be able to create um, organs, functional organs for individuals, instead of waiting for someone to donate an organ when they die. So that's where the future is going into. But this is particularly important in the case of neurodegenerative diseases. Now, I just named a few like Alzheimer's, Parkinson's, and Huntington's disease, because in case you did not know, your neurons don't regenerate. Um, Unlike many cells in the body, your neurons, they don't go through the cell cycle multiple times. So eventually, if you do lose your nervous tissue, for example, in these diseases, they don't grow back. So we'll speak about that a bit. So the first thing I want to talk about is Alzheimer's disease. In the case you did not know, Alzheimer's disease is a progressive and irreversible brain disorder that affects memory, thinking, and behavior. It is important. It is the most common cause of dementia, accounting for 60 to 80% of dementia cases. So like I was saying a while ago, nerve cells do not regenerate since they do not undergo numerous rounds of the cell cycle. So in Alzheimer's disease, the gradual degradation or degeneration, of nerve cells in specific regions of the brain leads to a loss of connections and communication between the brain cells. So other hallmarks of Alzheimer's disease includes amyloid plaques. So these are protein deposits that accumulate between the nerve cells and thus disrupts their function. And in the nervous system, it is very important that the synapse, which is a space between nerve cells, is kept clear of any proteins, because that is the area where neurotransmitters will come from one neuron to another to signal a downstream action. So if we have proteins being accumulated in that area, the neurotransmitters cannot interact with the postsynaptic. The presynaptic neuron cannot interact with the postsynaptic neuron efficiently, and then that's when you get all the symptoms seen in Alzheimer's. There's also neurofibrillary tangles. This consists of toll proteins that form inside the neuron, or they interfere with the transport system. So in this case, it's more to do with inside the neuron. Like a while ago, I mentioned the neurotransmitters. Those aren't created at the synapse, so those will be created in the cell body, and then they're transported down the axon to the bottom of the neuron, where they could then be transported through the synapse to the postsynaptic neuron. I should have put a picture showing the synaptic cleft so you have an understanding of how it works, but I'm hoping that you could visualize it. Once you know what a neuron looks like, you could visualize one neuron to the top and another one at the bottom, and the space between them, because they're not attached to each other. There is a space in between them. That is the area where the transmitters would pass through. I think I could draw. Yeah, I'll try my best to draw it. Let's see my artistic skills. Yeah, so I'm trying my best. So this is the cell body, this is the axon, and this is the bottom. And then you would have another neuron here. So this is your presynaptic neuron, this is your postsynaptic neuron, and this area here is the synaptic cleft. So what happens is the neurotransmitters would come from this neuron and it would travel down the cleft to the postsynaptic neuron. So if we have proteins in the area, then that would disrupt this transmission and, well, obviously affect the relay of information. Oh, let's take it to the top. But moving along, the application of stem cell technology. One moment, let me erase this so it doesn't distract you. Okay. So the application of stem cell therapy in regards to Alzheimer's. So although Alzheimer's disease is the most common form of dementia, there is no available cure available. Several research projects exist, but none is on the market at the moment. Stem cells, but one route that we could take is utilizing stem cells. Stem cells are unique cells with the ability to differentiate into various specialized cell types. So they can self-renew and repair damaged tissue, making them invaluable in regenerative medicine. So stem cell therapy aims to replace these lost neurons by newly generated functional ones, restoring cognitive function. So stem cells are like precursors. They're not yet differentiated into any specific tissue. Um, However, when they get certain signals, that's when they'll begin to differentiate into specific tissue. So this therapy aims at creating stem cells that would differentiate into neurons and replace the ones that were damaged and led to the development of Alzheimer's. So neural stem cells are a type of stem cell that can give rise to different types of brain cells, including neurons and glial cells. And harnessing the potential holds hope for addressing Alzheimer's effects. Stem cells can secrete neurotrophic factors, supporting the survival and growth of existing neurons. So this promotes the creation of a supportive environment for neurons to thrive. Additionally, through a similar mechanism, it regulates amyloid beta and tau proteins, which contribute to the formation of plaques and neurofibrillary tangles in Alzheimer's disease. So that's it for stem cell therapy. I just gave an example of a neurodegenerative disease, but you could imagine that this technology could be used for various disorders, various conditions. For example, creating skin grafts, using stem cells to create skin grafts for people who are burnt. But another thing we can do is actually start 3D bioprinting. And I'm not sure if you're familiar with it, but there are 3D printers all over the world right now, just for printing like physical objects. But now we're looking into 3D printing organs that could work. And I have a video showing one of the organs created at a university. I believe they created a functional heart. So we're going to look at that in a bit. But 3D printing, also known as additive manufacturing, has transcended traditional manufacturing processes to revolutionize the field of medicine. So one of the most groundbreaking applications is the ability to create functional human organs through 3D printing technology. And how do these the 3D printing of organs work? So you could have layer by layer fabrication, which is 3D printing builds structures layer by layer using biocompatible materials, so living cells and biomaterials that mimic natural tissue. In bio 3D printing, cells are combined with bioinks. These are hydrogels or materials to create intricate structures that mimic the architecture of real organs. Tissue maturation, printed structures are cultured and in a controlled environment to allow cells to grow, differentiate, and become functional tissues. So current applications, bioprinting skin. So 3D printed skin grafts are used for burn victims, helping to promote healing and to reduce scarring. Cartilage and bone. Bioprinting is used to create cartilage and bone grafts, benefiting orthopedic procedures. Heart tissues, researchers are bioprinting heart tissues for drug testing and studying heart diseases. Functional liver models are being developed to aid with drug metabolism studies. Now, for this one is very important for drug testing because a lot of times ethically we can't trust test drugs, on humans right away. Like I said, like it has to go through clinical trials, but this could speed up the process because right now you could 3D print a heart that replicates a human, like a human's heart, and you could test your drug on it without worrying about killing the person because um, you it's not a living like heart. It's not from a living individual. You didn't take it from someone. So also, so that opens up some great um, pathways for drug testing. So this is the video I had. Let's play it. To screen. So it's a bit slow.
And when it comes to printing human body parts, the future is already here. Just in the last few months, we've seen some pretty incredible results. One woman received a 3D printed ear implant made from her own cells as part of a clinical trial. Doctors in France were able to grow a new nose for a cancer patient using 3D printed cartilage. And get this, researchers at MIT can now print 3D replicas of the human heart.
The team has developed a robotic system to control soft 3D printed replicas of a patient's heart that can be actuated to mimic the patient's blood pumping ability. The procedure involves first inverting medical images of a patient's heart into a three-dimensional computer model, which the researchers can then 3D print using a soft polymer-based ink. The result is a soft, flexible shell in the exact shape of the patient's own heart.
Wild, right? Anytime I see something like that, I call NBC News medical fellow Dr. Akshay Sayal. He joins us now. Doctor, why does this matter? What implications does this have on the future of organ transplants?
Yeah, good evening, Gotti. The simple way to put it is we do not have enough organs out there for all the people who need organ transplants. Every year, there's about 100,000 people on the organ transplant list, and about 17 people die every day waiting for an organ transplant. So that's why you're seeing a lot of excitement about this. We really need a way to solve that shortage of organ transplants, for patients who have kidney disease and heart disease and liver disease. You and I spoke a few weeks ago about a patient who needed a heart transplant who almost didn't get one. So that's really why you're seeing the excitement about this, growing an organ in a lab as opposed to waiting for an organ to become available.
And how does this actually work? Explain the process from design and printing. We saw a little bit of the printing there to physically making a 3D organ part for the human body.
Yeah, it's pretty complicated, but I'll try to break it down here. And basically, what you do is you take a piece of tissue sample or a little piece of sample of an organ that you want. Say you're in need of a kidney transplant. You can take a piece of that about the size of a postcard, and you mix that with a bunch of fancy chemicals, and then you put that into literally a 3D printer. So if you think about a color printer that uses ink to combine a bunch of colors together to get the final product, you're taking a bunch of cells and you're kind of mixing them together and combining them together, and then the 3D printer kind of layers them on top of each other until you literally get a 3D printed organ. But Gotti, I think the most important point here to drive home, because this is coming from a patient's sample, you don't have to take those immunosuppressive medications, those medications that prevent organ transplant rejection, that can actually weaken your immune system and make you more likely to get sick from things like COVID.
I was expecting like an hour-long answer, and I was ready for it. I was getting ready to sit down and listen to all of it.
Yeah, sorry about that. Let's go back to it.
And when it comes to those medications that prevent organ transplant rejection, that can actually weaken your immune system and make you more likely to get sick from things like COVID.
I was expecting like an hour-long answer, and I was ready for it. I was getting ready to sit down and listen to all of it. I don't know how you just summed that up in like 2 minutes. I want to know though, how is this even regulated? This seems like a very brave new world, and how far off are we from seeing this become a huge thing that doctors and hospitals are using on a daily basis?
Yeah, so the experts really at Wake Forest who are leading the charge of this, they think about 10 years out is where we are right now. And if you guess, the organ we're most in need of a transplant for right now, it's actually kidney disease. I think it's about 90% of patients who are in need of an organ transplant are waiting for a new kidney. This is like you see on the screen here, about 100,000 people waiting for an organ transplant. But to answer your question, we don't really know how it's going to be regulated. I think that really the important thing to drive home is we need to figure out ahead of time how we're going to get this to people who need it most. Because when we go back to the kidney, the people who need it most, over 50% of those waiting for a kidney are minority populations. You want to make sure they are included in talks to get access to this as well. And I just want to wrap here, Gotti. I know I said this is 10 years out. For all those out there watching, it's not too late to be an organ donor. If you can, go to www.organdonor.gov. Sign up today if you haven't already.
Dr. Akshay Sayal, every time I talk to you, you blow my mind. Thanks so much.
Anytime.
All right, so this was just a summary on the technology. Like you said, it's not yet mainstream. So, but right now we're just testing out the technology, and that's a lot of things that's ongoing in biotechnology is really testing it out to see like it actually works, and then eventually wait on the organizations to come up with regulatory regulations on how we're going to use this technology. But I don't think that should limit us on exploring it. One important thing that he said, I'm not sure if you took note of it, to also lean on what we did this morning in immunology, is that because we're using the cells from that individual to create the organ, they don't need to consume any immunosuppressants. So usually if you donate an organ, so if you receive an organ from someone else, that person's organ is made up of their cells. So their cells would have the antigen, that antigen that's displayed on their cells would seem to our immune system as foreign, because it has a different self-antigen. It's not the same because we got an organ from someone else. So our immune system would recognize that, and it would immediately attack it. So just like we spoke about this morning. So people who get organs from donors, they need to take immunosuppressants, which would lower down their immune response so that they could survive. Because if they don't take those pills, what would happen is the immune system would just destroy the organ that they just received and render it useless. So that is why, and these people who get organs, they have to take those immunosuppressants for their entire life. So it shows you how this technology could benefit individuals. However, like you said, we are years down the line of it being mainstream, but it just shows that we have the technology available to us, and it's being optimized and becoming more efficient day by day. But yeah, the next thing we wanted to get into is genetic engineering, and speaking about GMOs, and a lot of ethical questions arises in the genetic modification of organisms. That includes crops and animals, and right now also humans. Considerations include the potential impact on the environment, the biodiversity, and unintended consequences on the ecosystem. People are opposed to GMOs because they believe that they are dangerous to the environment. They come with unknown effects on human health. They reduce biodiversity, lead to the evolution of resistant pests. However, the utilization of GMOs can allow for the development of disease-resistant plants, plants that are capable of surviving climate change, plants with bigger yields. In our case, I'm just speaking about theory-wise, we could also cure diseases or make changes that could change the lifestyle of individuals. So in yesterday's sessions, when we went through those genetic disorders, we saw how having a faulty CFTR gene could lead to the onset of cystic fibrosis. But what if you do a genetic test and then you use the technology like CRISPR-Cas9 to edit that out and replace it with a healthy genetic code, then that person does not get cystic fibrosis. So that is just theory of what we could do, but obviously there are a lot of ethics involved in this topic. So this is a video on GMOs. This is about 9 minutes speaking about them. So we're going to see that, watch it.
GMOs are one of the most controversial areas of science. Genetic engineering is used in many fields, but even though medical applications like GM insulin are widely accepted, the debate heats up when it comes to food and agriculture. Why is that? Why is the same thing treated so differently? Let's try to get to the bottom of this and explore the facts, the fears, and the future of GMOs. Humans have been genetically modifying plants and animals for thousands of years. Maybe a few of your crops had very good yields. Maybe one of your wolves was especially loyal. So you did the smart thing and bred the plants and animals that had traits beneficial to you. Traits are just an expression of genes. So with each generation, those genes got more pronounced. After thousands of years, almost every single plant and animal around us is vastly different from its pre-domesticated state. If humans have been changing genes for millennia, what makes a so-called genetically modified organism, or GMO, different? Selective breeding is basically hoping for lucky hits. Genetic engineering eliminates this factor. We can choose the traits we want, make fruit grow bigger, immune to pests, and so on. So why are people concerned about them? Let's start with one of the most common objections to GMOs. Gene flow, meaning GM crops could mix with traditional crops and introduce unwanted new characteristics into them. There is a method that might guarantee complete prevention, but it is a big anti-GMO argument by itself: terminator seeds. The idea is that they could produce sterile plants, requiring farmers to buy new seeds every year. The very concept of this, however, caused a public outcry, stopping the technology being put to use. This brings us back to the unintentional spreading of engineered DNA. There have been cases of GMOs growing where they weren't planted and traces of modified genes found in foreign crops. But GM plants can't run wild entirely. Many crops pollinate themselves, and all crops have to be related to mingle. There are also cultural methods like buffer zones to keep unintentional crossing at a minimum. But if it's possible in principle that a GMO could unintentionally cross with a non-GMO, there's actually a more important question. Is food that comes from GM crops different to food from non-GM crops? This question has been a major concern from the very beginning. GM plants that are destined to be eaten are checked for possible dangers, and the results are evaluated by multiple agencies. After more than 30 years and thousands of studies, the science is in. Eating GMO plants is no more risky than their non-GMO equivalent. But don't just take our word for it. Sources for this and other claims are in the video description. But what about plants that have been engineered to be toxic? For example, BT crops. A gene borrowed from the bacterium Bacillus thuringiensis lets engineered plants produce a protein that destroys the digestive system of specific insect pests. The plant makes its own pesticide. Insects that eat it die. That sounds alarming. Pesticide sprays can be washed off, while the poison in BT crops is inside the plant. But actually, it's not a big deal. Poison is really just a question of different perspectives. What's harmless to one species might kill another. Coffee, for example, is a poison that kills insects, but is harmless to us. Or take chocolate. It's dangerous for dogs, but a pleasure for humans. BT crops produce a protein that is tailored to the specific design of the digestive tract of certain insects. It's completely harmless for us. There's also the opposite approach. plants that are engineered to be resistant to certain weed killers. This way, farmers can use them widely, killing the other plants competing for resources without harming the crop. Here we get to the dark underbelly of GMOs. For the pesticide industry, they are big business. Over 90% of all cash crops in the US are herbicide resistant, mostly to glyphosate. As a result, the use of glyphosate has increased greatly. That isn't only bad. Glyphosate is much less harmful to humans than many other herbicides. Still, this means farmers have a strong incentive to rely on this one method only, casting more balanced ways of managing weeds aside. That's one of the most fundamental problems with the GMO debate. Much of the criticism of this technology is actually criticism of modern agriculture and the business practice of the huge corporations that control our food supply. This criticism is not only valid, it's also important. We need to change agriculture to a more sustainable model. GMOs as a technology are actually an ally and not an enemy in that fight, helping to save and protect nature and minimize our impact on the environment. Let's look at some positive examples. Eggplant is an important crop in Bangladesh, but often whole harvests are destroyed by pests. Farmers had to rely heavily on pesticides. Not only was this very expensive, farmers also frequently got sick. The introduction of a new GM eggplant in 2013 stopped this. The same Bt protein we talked about before, an effective killer of insects but harmless to humans, was engineered into them. This reduced insecticide use on eggplants by more than 80%. The health of farmers improves and their income rose dramatically. And sometimes a GM approach is the only option. In the 1990s, the papaya industry in Hawaii was under attack from the ring spot virus, which threatened to wipe out Hawaiian papaya. The solution was a papaya genetically modified to be vaccinated against the virus. Without it, the state's papaya industry would have collapsed. All these stories show a very narrow application. 99% of all GMOs we use right now produce pesticides or are resistant against them. There is so much more we could do. Scientists are working on GMOs that could improve our diet. Plants that produce more or different nutrients, like fruit with higher antioxidant levels that help to fight diseases, or rice with additional vitamins. On a larger scale, we're trying to engineer plants that are more resilient to climate change. Plants that can better adapt to erratic weather and adverse soil conditions, making them resistant to droughts or floods. GMOs could also not only reduce agriculture's impact on the environment, but actively help to protect it. Scientists are working on crops that can draw nitrogen from the air like microbes. Nitrogen is a common fertilizer, but its buildup pollutes the groundwater and speeds up climate change. Plants that collect their own nitrogen can fix two problems at once: the overuse of fertilizers in the developed world, as well as the shortage of it in developing countries. We could even modify plants to become super effective carbon collectors like the American chestnut tree to mitigate and actually reverse climate change. With the tools we have today, our imagination is the limit. The world eats 11 billion pounds of food every day. A UN estimate suggests we'll need 70% more by 2050. We could grow that food by clearing more and more forests to create fields and pastures and by using more pesticides. Or we find a way to do it on the land we've got right now with more effective methods like GM crops. Intensifying farming instead of expanding it means that GMOs could become the new organic. In a nutshell, GMOs have the potential to not only drastically change agriculture, but to also dampen the effects of our own irresponsible behavior. GMOs could be our most powerful weapon to save our biosphere. This video took more than 600 hours to make.
All right, so this was a summary on GMOs, the plant version. If you go on YouTube, you could also get one speaking about us engineering the genome of humans. But I wanted to show the plant version particularly because I know an argument out there right now is that eating GMOs is not good for you. And yeah, so people have that stigma against it, but what we don't realize is that a lot of the crops that we eat right now that we deem natural have had their genome edited from years ago, the tomato that you know now to be that ripe plumpy fruit, that's not what it was before. The original tomato that was found in I believe South America was very small, and it was until that we crossed it with other members of the family that we were able to create that plumpy tomato that you know now. And we've also made it so that it lasts longer on the shelves. So the issue with the GMOs, I believe, like you said in the video, is really around the economic side of it, being like those big industries controlling those crops and it not being in favor of the smaller farmers. But in regards to the technology itself, from all the research that they've done, there isn't really much wrong with consuming genetically modified organisms. And it's really important for the future, especially with climate change. One of my lecturers in Trinidad, because in Trinidad I did plant genetic improvement as a course, and my lecturer there is actually doing some research on how they could make some cocoa plants that could withstand the extreme heat and also lack of water. So that's what they're working on right now in Trinidad, because Trinidad is one of the suppliers of a lot of cocoa globally. So that is a pretty promising research. I can't wait to read the paper when he actually publishes it.
Which would be impossible without viewer support on patreon.com. If you'd like to support carefully researched content made with love, it's really very helpful.
Not sure why it started playing. Let's just continue. So now we're going into the human genome editing. So the human genome editing involves precise modifications of the DNA within a person's cells to treat or prevent diseases. This powerful technology has the potential to reshape the landscape of medicine and genetics. The application of human genome editing, so in genetic disorders, so for example, the ones we spoke about yesterday with Tay-Sachs, sickle cell, cystic fibrosis, genome editing can correct or mitigate genetic mutations responsible for diseases like cystic fibrosis or sickle cell. Cancer treatment, editing immune cells to be better, like to better target cancer cells and showing promise in immunotherapy, infectious diseases, editing cells to make them resistant to viruses like HIV could revolutionize disease management. Personalized medicine, tailoring treatments based on a patient's genetic makeup could improve efficacy and reduce side effects. So the future is bright when it comes to human genome editing. However, there are some challenges and obviously the ethics side of it. So some challenges. So while biotechnology offers immense potential, it also faces challenges that include high development costs, regulatory hurdles, and access to treatments. So ethically, you have to consider germline editing. So editing genes in embryos or germ cells raises ethical questions about altering the human germline. Unintended consequences, off-target effects could introduce unintended changes to the genome, potentially leading to new health risks. And that is true because we still don't know a lot. Like, yes, we've established what our genome is made up of. We don't know what change in one gene does to another because a lot of the genes of our body are interconnected. So these are all things that we need to consider when making those changes, if that ever happens. Inequity and access, ensuring equitable access to genome editing therapies raises concerns about socioeconomic disparities. This technology, yes, it's effective, but it's not cheap. It will be expensive. So then now you have to think of who gets access to that technology, who could use that technology. Will the billionaires and millionaires be the only ones who can save their children? Will they use their money to assist persons suffering from those disorders? So these are things that need to be considered. And then there is also the possibility of designer babies, quote unquote designer babies, which is the prospect of creating designer babies with enhanced traits raises ethical dilemmas. So people now want to, if we start like making that technology accessible to everyone, we'll have people who want to change everything about their child. So you want your child to be 6 feet, you want your child to have blue eyes, you want your child to have curly hair, light skin, and you're dark skin. So these are all things that need to be considered. That's why it's very important that there is like regulation of the technology. And for those reasons, there is this debate in the scientific community on whether we should rule out like making changes to the genome, or if we should just let it be natural. I personally stand on the point where it should be accessible, however, strictly regulated, because I believe that if we could change lives in regards disease-wise, I feel like it's worth the risk, because someone suffering from sickle cell lives a completely different life from someone from a healthy individual. So if we have the technology to make those changes, I don't see why we should not use it. And this is just an interesting story about a doctor, Yi Jiankui. I'm not sure if you've ever heard about this doctor before, but he used CRISPR, which we will speak about when we talk about DNA technology. But the ability to edit human genes using techniques like CRISPR raises ethical dilemmas. Debates focus on issues such as potential for germline editing, the same thing we were speaking about, safety concerns and concerns about creating designer babies. So more or less reiterating what I just said. But this doctor became the first scientist to create genetically modified babies using CRISPR-Cas9. In his experiment, he fertilized a sperm and an egg in vitro. And after fertilization, he was able to make edits to the genome. And the edits he made were to a gene that codes for a protein HIV utilizes to enter the cells. So what he did was, he had no permission to do this. He did this in his own laboratory. He went ahead and he made those changes to those twins. And what happened was that initially the response he got was good. Like everyone was praising him because now these twins could be resistant to HIV because the gene that codes the cells that HIV enter is no longer in them. But then he got a lot of backlash for doing that, and he was sentenced to jail for 3 years. He only came out last year out of jail. I don't think he still has his license to operate. So yeah, so that just shows how the world is in regards to making edits to the human genome. But that concludes my presentation. It's something you could read about his story and his experiment. Yeah, I'm not sure if anyone has any further questions. Okay, so if no one has any questions, that's where we stop for the day. Giovanni said that's cruel. Well, the thing is, you need to, once you're in the scientific community, like once you're in the science world, like you need a lot of approval for these things. You can't go ahead and just make changes because that is a human being. Right now, yes, there's the editing made them resistant against HIV, but you don't know if those edits would have caused something else down the line. So like these children are constantly being monitored to this day to find out if there are any other like adverse effects. Yeah, so but yeah, I understand how you say it's cruel, but at the same time, it literally just changed the lives of those individuals. And we're in a space where we don't have like regulatory criteria met as yet. Like nothing has been established. Like the technology is just growing. However, there is potential for the next 5 to 10 years. But yeah, I would like to say thank you for giving me your attention for the last 2 hours. I hope you guys have enjoyed the rest of your day, and we'll continue tomorrow to speak about DNA technology and also more biotechnology.